o'clock. So we're going to get started. Welcome everyone to the first Sonic Speaker event of 2016. We're very excited to welcome Dr. Charles Macal of Argonne National Laboratory. Dr. Macal is a global leader in the growing field of agent-based modeling, and he's also an alumnus of the Northwestern Industrial Engineering and Management Sciences program, and we're happy to have him back. He's going to tell us all about chiSIM, or the Chicago Social Interactions Program, and its applications across several different disciplines. So, without further ado, take it away, Chick.
Okay, thank you, Megan, for that wonderful introduction and for setting up the seminar today, which was basically a year in the making to get the dates right and things like that. I'm very happy to be here at Northwestern. Again, being an alumni, and later today talking to some people from the department. I'd like to thank Nosh Contractor for inviting me to give this talk as well. And as Megan said, I am Charles Macal, or my nickname is Chick. I've had that my entire life, Chick Macal, and I'm from Argonne National Laboratory. And Argonne is located on the southwest side of Chicago. Perhaps some of you have been there or have attended some of the open houses we have, things like that. It's a Department of Energy owned facility, and we do a lot of research in the field of energy and environmental analysis. Things like national security as well, and basic scientific research with our various types of user facilities like exceptional large computers and accelerators of various kinds to do various kinds of physics experiments and applications. But we also do many other things that you probably wouldn't expect the National Laboratory to do. And these include things like applying computer science and computation and modeling in particular to problems that are broadly of national interest in one way or another. So we do work on things like climate change, and some of the things that are part of that are things like modeling society or human behavior, and other things that have traditionally not been the realm of physical sciences and engineering disciplines. So that's kind of the backdrop for my talk in terms of where we are coming from. I want to say, Uri, yeah, hi. I do want to also introduce a couple of colleagues that I have brought with me from Argonne, because the work that I'm going to be doing and sharing with you, I should say, and describing, is basically a team effort of our interdisciplinary research teams and collaborations that we have going on at Argonne. So first, and I brought them with me today. I apologize for having to have them sit through my talk. I've done that before. Apologize earlier. But throughout the day, then we're going to have meetings and talk to people here at the university, and hopefully, develop some kind of ongoing interactions and even collaborations and things like that. So anyway, I'd like to introduce Jonathan Ozick. Dr. Ozick is working in the area of computational social science. And Dr. Jessica Trail is working in the area of applied statistics and modeling at Argonne. So with that introduction, I'm just going to jump in to the talk. The talk is titled Simulating Chicago and Everyone in It. So the basic outline is just a few words about agent-based modeling as an introduction for people who maybe aren't familiar with that. I do want to point out that I'm well aware that Northwestern is one of the the Center for Connected Learning is one of the primary places in the world for agent-based modeling, platform and software development. So just want to acknowledge that. I'm not going to tell you anything new there. In terms of, then the other part of the talk, we'll talk about synthetic populations and the development of one for Chicago. that we have developed and are continuing to improve upon. I'll explain the structure of our model, the Chicago Social Interaction Model, or chiSIM for short, and then just get into an application that we've done for community-associated MRSA modeling, for Chicago on a funded project by the NIH. And then finally conclude with research, interesting areas for research challenges. Okay, so a few words to begin about agent-based modeling. Agent-based modeling is an approach to modeling and simulating systems comprised of autonomous interacting agents. Those are probably the key characteristics, those two, autonomous and interacting. Often agents represent people. They could represent other things like animals or organisms, things like that, even organizations. Agents have behaviors, and that represents a challenge for how we model behavior in particular for modeling people. The key outcome has to do with the dynamics, and how the system changes and evolves due to the behaviors and interactions of the agents. So agent-based modeling is a very forward-looking, dynamic technique that is capable of modeling and including things like turning points and tipping points, and those kind of effects that you wouldn't normally expect to get out of, let's say, a linear type model because of the nonlinearities that are introduced by the interactions of the agents due to their behaviors as well. Agents are interacting. They're typically receiving information and interacting with others in a local area or neighborhood. The basic idea is everyone doesn't interact with everyone all the time. So in that sense, it's somewhat different than things like molecular dynamics or modeling molecules, which have force influences throughout the entire space. And the primary reason we do agent-based modeling is to determine what, where, when, and how macro or system level characteristics and consequences emerge from the micro level or individual level behaviors of the agents. So because these approaches, well, the kind of models that we're looking at or including, the details we're including into models are not tractable by analytical means normally, then the necessity is to resort to computation to simply find, so computing the model is like finding the solution of a model if we had analytically. So a way to think about agent-based modeling is to think about the agents in the model and what their attributes are and their behaviors. So typical attributes of an agent would be the name for the agent and several static attributes, perhaps socioeconomic variables, gender, household structure, etc., as well as attributes that are dynamic, that would change during the course of the simulation. So perhaps we might model an agent as having experiences within the model from its interactions, perhaps acquiring a disease in our model if it's a disease model, and recording that fact into their memory. And the memory would be updated as more interactions occur and events occur. Agents also could have resources, whether it's money or a store of information, as well as networks of other agents through which interactions occur. Behaviors convert sensed information into agents' action in the model. And so basically, the behaviors allow the agents to be autonomous. And we don't have to tell the agents what to do in the model. We just give them their behaviors and set them off into a simulation framework to go to the future. In addition, behaviors are based on an agent state, which consists of whatever the state is of interest. If it's in a disease model, it could be whether the agent is infected or simply susceptible or possibly recovered, for example. And then the agent behaviors are then based on what that state is. So an agent may have a different behavior if it's infected versus if it's susceptible. And we want to model that some course in the simulation. And then finally, one of the interesting aspects of agent-based modeling is that it allows us to explicitly model learning on the part of the agents. Agents are then able to either learn new behaviors or adapt their behaviors to new situations that they haven't encountered before. So these agents then interact in what I call a social interaction space, which is defined, let's say at the most sophisticated level, through networks that indicate which agents interact with which. And depending on the application, those networks may be created or, let's say, dissolved over time for various reasons. And in some of the work we're doing on HIV type modeling, we're modeling HIV networks of young men and empirically estimating how those networks grow and then how they over time dissolve based on the relationships that we see in the data. But anyway, I did want to just very briefly mention the other kinds of applications of agent-based modeling. Agent-based modeling is somewhat of an unusual kind of, let's say, modeling technique, because it has this enormously broad applicability across a wide range of disciplines. For the most part, many disciplines for which simulation has never really been widely applied before. And so at Argonne, these are just a list of our Argonne large-scale agent-based models that we've done or are under development. And as you can see, they cover everything from application areas such as supply chains and markets, technology adoption, such as solar photovoltaic technologies, things like that, which you might imagine the Department of Energy is interested in, learning about, to the environment, as I mentioned earlier, including global climate. Healthcare and infectious diseases is a huge area for applications of agent-based modeling. There's many funded programs for that because it allows one to model things in the area of healthcare and infectious diseases that are simply not able to be modeled by any other, either simulation or even analytical approach, to at least get some insights onto how systems may develop in the future. But probably more importantly than anything else is to see what the effects of interventions are, either government policies or events that may occur in the future upon the systems that people have to manage. Biology is another area at the lower level of agent resolution, so to speak, where cells actually are agents. And then social cultural modeling, where probably was kind of the real initiating field for the motivation of kind of the surge in agent-based modeling was through the of the last 20 years the models of social systems and social behaviors and things like that, which led to in part the entire field, of computational social science, which includes agent-based modeling, but a whole lot more. Okay, so on to chiSIM. Okay, just imagine a new generation of analytical tools for understanding the dynamics of cities, for managing cities in terms of design, planning, operations, and tools that are continuously calibrated with real-world data and validated. So here's what we have is a model of Chicago as viewed from the south side, where I spend more time on the south side than the north side, because that's where Argonne is. And typically, architectural design firms, for example, this is actually a figure that was created by Skidmore, Owings, and Merrill, which we worked with on adding agent-based modeling into their repertoire of tools. In any case, this is Chicago. And so, the question is, what is missing from this picture? Right? When we design cities, when we understand cities and the people in them, well, shouldn't say that, shouldn't give it away. When we design cities, the question is what's missing from the normal picture of design. Oops. Well, the answer didn't come up, but the answer is people. Sorry, I had an extra slide in. Okay, so the approach that we're taking is to add people. One way to look at it is to add people into the city, or add people into the built environment. And that can go on for a variety of reasons. But basically, here's how you would actually do that. These are the 4 steps that we've come up with. First, create a synthetic population of virtual agents. And these consist of things like people. But they also consist of places as well. And they can consist of other structures, such as households that are collections of people and things like that are very important for modeling how people behave and decide how to do things. Secondly, mobility. We'd want to ask, where do people go and when? And what the prospects are for co-location of people, such that they can interact in some way and have social interactions, so to speak, whatever those may be. But another aspect of mobility has to do with people's activities. So we'd like to know, what are they doing when they are located at these places? Are they, for example, in modeling disease propagation, we'd like to know what people are doing if they're in a particular location that could perhaps promote transmission of disease. So we can do different things at different places. And that's an additional piece of information that one may not necessarily get just from these rampant mobile phone studies and things like that, where people are actually located. So finally, not finally, but we'd like to add behavior in terms of how people are socially interacting and how this results in information diffusion, what people learn from each other, potentially, and the reactions of people to that information as well, in terms of allowing them to make decisions, facilitating the process. In the end, the objective is to be able to model interventions and do what-if analysis for, as I mentioned earlier, government policies or other public policies, to see what their reactions could be if considered at an individual level. And then, in addition, the final step is to model social interaction through either social networks, social contexts, and the kind of behavioral influence and decision-making that is actually suggestive of what goes on in the real world in terms of how people make decisions and who they get information from, and things like that for those decisions. So, in terms of synthetic populations, a population of agents in an agent-based model is referred to as a synthetic population. And generally, one can construct a baseline synthetic population that includes the general characteristics of agents and their behaviors, including socio-demographic data, as well as their daily activity set. But these have to be assembled from the ground up and from many data sets. So we're seeing a kind of just now multiple research organizations producing synthetic populations for people in various places, around the world. But these are of a general nature. In fact, I'm aware of some activities going on in which one group at Virginia Tech is producing synthetic populations for the entire world. And the reason is because They are being asked to look at infectious diseases, these emerging infectious diseases around the world. So they have to be ready to create an agent-based model of any place in the world for which an infectious disease outbreak occurs, and to be in a position to do some analysis, on that. This has come up in particular and spurred on by the Ebola outbreak, last year, well, 2 years ago, leading into last year. And now the Zika virus outbreak is kind of, organizing everyone around that as well. And there are many people, many governments are involved in, trying to understand those efforts. So there's, of course, there's probably, this is not a joke, but there's efforts underway to develop synthetic populations of mosquitoes as well by species around the world. And that would be valuable information to know. Okay, so in addition to general data sets, we have specialized data sets, which are required to supplement the general synthetic population for any particular application. Because every application one does requires simply data sets that are only available for particular locales, or that look at attributes that are simply not available in the general data set. And I should say that in the general data set of synthetic populations, one begins with census data and other things that are generally publicly available, and supplements it by special collection methods for additional items. So in developing, let's say, the model for Chicago, we supplemented the data which came from the census with specialized, attributes and variables that we needed, not only for Chicago, but also for our application in modeling infectious disease propagation. So basically, the agent-based model then is populated with agents that reflect the same distribution of individual characteristics as the corresponding population of people in the real world, but only at the aggregate level. So typically, when synthetic populations are put together, there are not as many anonymity concerns, in the sense that no single agent represents a particular person necessarily. But it's only statistically, when one adds it up, we get the same distributions, no matter how that cross-tabulation is done. Another part of synthetic population development is to do assignments of people to places and activities, as I mentioned. So far we've done that through algorithms, where some of the examples are people are assigned to households, students are assigned to schools, people are assigned to workplaces, et cetera, based on various algorithms that have been developed. Some of them are available in the literature and peer reviewed and things like that, to make those matchings occur. In addition, there are all kinds of other places generally called group quarters, including things like college dormitories and nursing homes and things like that require special treatment. And finally, some group quarters, and this is very interesting, like prisons or jails, nursing homes, are also places where you want to assign workers. So there's an additional aspect of people going to these places, say students going to school, but also the need to assign workers and teachers for those schools as well. And within that context, these people have different contact patterns, behaviors, things like that. So in other words, creating a synthetic population requires linking together many synthetic population data sets and the attributes that are available from different sources. So here, essentially, in the end, we boil it down to 3 kinds of files or data sets. One has to do in the middle here with people. It's a people file. So there's an entry for every person in the synthetic population. In the case of Chicago, for example, we have, depending on, you know, how you define the region, we have either 2.9 million agents, one per every person in the Chicago city limits, or we have 6 million, depending on whether we're including Cook County and DuPage County as well. And, you know, larger areas would give us more for the entire metropolitan area. But in addition to people, we have all the places for which the people are associated with. So in Chicago, for example, there's basically in city proper, there's 2 million places, which isn't that many when you think about it. And so we construct a file for all the places, which includes the kind of place it is and the specific latitude and longitude, because these are actual real places that we're looking at. And so we can link the persons to the places through what places people go through or to in their normal daily schedules by linking in basically an activity file as well. So we've taken data from the American Time Use Survey, which is a survey of what people do that's recorded every like 11, I think it's, I don't know, every minute basically that people change their activity, they write in some diary, you know. So there's these huge files and they're all grouped by socio demographic characteristics, you know, or at least indexed by that. So basically what we can do is using that data for adults combined with data for children who are under 16 or 17, which is called the panel, the survey of income dynamics, panel survey of income dynamics. We can get a full characterization of the entire population in terms of what activities are. Now these are national surveys, of course, so we basically group activity profiles by socio-demographic characteristics and would just, you know, kind of match clusters of activities to actual people in our people's file and assign, therefore, a whole set of activities that a person would go through with those same characteristics throughout their entire day. So what this allows us to do, and you know, this is very kind of complex data mapping and things like that shown there, is then allow us to actually see the schedules, synthetic schedules, let's say, for all the people in Chicago. And then we can step through those schedules to see the places people are going to. And then with the matching algorithms, we can match which particular places of that type people are going to. So we can finally get to, you know, representing a person, a child, let's say in a household in Chicago, and figure out what school, at least for the purposes of the simulation, they go to. And then simulate that movement process according to their schedule of when they're leaving home and going to school. Okay, so that kind of cross-matching and everything allows us to do some analysis. Let's say kind of pre-analysis. And here there's a couple of graphs that simply show various ways to cross-cut the data sets. So we have, yes, this is by day week. So take all the people in Chicago, well, in the simulated Chicago, and see what they're doing by every day of the week. We can plot out what the activities that they're engaging in for each of those days. And then in this case, we've mapped those activities to whether those are high risk or low risk relative to getting a particular kind of disease. In this case, it was a MRSA infection. Some activities are more conducive for transmitting MRSA. So if we were engaged in, if we were at a school or for children engaged in athletic activities at schools where they have a lot of skin contact, for example, it's a primary place for MRSA transmission, for example. And then similarly, just cross-cutting the database by another way, we're looking at white, black, and Hispanic by race. We can kind of look at the same kind of activities that each of these groups is engaging in relative to its propensity, let's say, or likelihood of transmitting MRSA, in case anybody has it, okay? So this is actually the probably the diagram that I have that best kind of puts it all together. But what we're doing within the chiSIM model then is to take all the data on people and places and combine it with activity profiles to arrive at co-location events that occur on a timeline. So at every place in Chicago, for example, we would know where what all the agents are at a particular time. Particular hour is our time increment actually in the simulation. And we have other algorithms then that take, let's say, the number of agents in a school at a particular time, a school location, and then stratify the agents at the school at that time into their age groups, because they would have more contact with people in their age group. But then we stratify, depending on how many students there are, we stratify them into like chunks to represent classroom sizes. Let's say you have chunks of 30. And then within that final group, we would kind of declare or model that there would be interaction possibilities of a higher nature for people within that group. And then we also have, like in the case of schools, we would have a possibility also that a child in 4th grade could interact with some child in 8th grade, as well, to a lesser degree. So the kind of social contact or social interaction, what actually would happen then is an additional element of the modeling process. So the example that I'm going to talk about has to do with disease propagation, as I mentioned, and that's kind of where the model actually arose out of, the problem of disease propagation. But anyway, just to give you an idea of the scale of the model, this is for Chicago proper. We have different places represented on the map here, including households, workplaces, schools, nursing homes, hospitals, gym, jail, and college dorm. These were the places of particular interest for disease propagation of MRSA. In particular, a strain called CA-MRSA, community-associated MRSA, which is USA 300. This is a different strain than the normal HA-MRSA, or healthcare-associated MRSA. CA-MRSA is much more virulent, and it's actually taking over as the primary strain in most places around the world, actually. So that was the nature of our research project, to investigate community-associated MRSA, which lives in the community, not only in healthcare settings. So that's why we had to develop a model for the entire community, just to see what the interactions were with various types of places that had been theorized as being particularly important as transmission centers for CA-MRSA. Thus, these are the places of particular importance. There was a theory that, for example, the jail, the Cook County Jail in particular, which is located in this area in several buildings, was a primary source of CA-MRSA transmission into the community. And so that was a theory that we had to investigate with our model. In addition, the question always came up about what are the role of hospitals, and more broadly, where are the primary places? So anyway, the scale is 2.9 million agents. Okay, 1.2, I highlighted earlier, it's 1.2 million places. We do the simulation on an hourly basis, and to reproduce the epidemic or buildup of CA-MRSA in Chicago, we had to do the simulation for 10 years at 1 hour increments. And each agent has a very limited number of behaviors, but nonetheless engages in mobility and movement in their daily activities, and they have contact with each other, that kind of thing. So if you add up all the contacts that occurred in the model between every pair of agents, it comes out to something like 3 trillion over this 10-year simulation. I'm going to kind of fly through this MRSA background, but community-associated MRSA is, as I said, a form of MRSA, and MRSA is methicillin-resistant Staphylococcus aureus, and methicillin-resistant just means that the bacteria has acquired some extra genetic information or capabilities such that it can resist penicillin or artificial penicillin, which is methicillin, attempts at it. It can be treated with other kinds of high-powered antibiotics, but the problem often is that the MRSA infection may not be recognized as a MRSA infection. Doctors may diagnose it as a spider bite or something like that, and then the disease can progress way down the road, and then maybe weeks later they're trying these high-powered antibiotics because they figured out that it was actually MRSA. It's spread by skin-to-skin contact, thus athletic activities, normal kind of things that occur in your household, people just touching and hugging each other, children with their parents, those kind of activities are normal, are how one can get MRSA as much as anything. It still does occur in the hospitals as well. And only about half of the skin and soft tissue infections are actually brought to medical attention. There's a very high risk of repeated infections. Actually, there's actually 3 disease states. Sorry, the disease states for MRSA are susceptible and affected, but not only that, there's one that's colonized, which is not shown here. So you could have MRSA living on your skin and be perfectly healthy and not know it. But you can infect yourself if you have a lesion or some type of injury. And so people, just playing athletics could get some kind of bruise or something, skin abrasion, and infect themselves. Now, the first site of the infection would be on their skin, but then if it got in your bloodstream, it could cause severe, infection and possibly up to death. So what we've done is we've modeled the disease states that are possible of individuals in the population. So we've kind of attached that to the activity simulation in which we've established contact for people. And then with a certain probability, and a colonized or infected individual in the same place at the same time can transmit that infection or colonization even, to another person in that same location. So that's the idea of the model. The theory behind this is well-founded, and it's very simple. Contact leads to transmission with a certain probability. In addition to that, though, there are other disease state transitions where a person who's colonized can simply, their own bodily defenses can fight off the infection or even the colonization. And then a test in a few months would reveal they have no colonization. They've decolonized themselves. Yet they can spontaneously colonize themselves as well. So there's this kind of battle going on in your one way to think about it, on your skin, of all these different strains of bacteria. And staph, which is not necessarily resistant to methicillin, is also present on your skin. And it's battling with the other strains of staph. So anyway, it's a complicating thing, and there's very little known about, because we'd love to model that kind of process within the individual. There's a strain competition, we call it, but we don't really know, there's not enough knowledge how to do that. But I guess we could still try. Anyway. This is basically how, so I'm just kind of putting all this together. This is how the model actually works. At the very top, we have initialization of the model at time 0, of course. And then agents remain in place, or they move to the next place for every hour. These are places. But in particular, there's two places that we need to focus on in more detail, and those are the hospitals and the jails. So we don't treat the hospitals and the jails as just little boxes that people go into, but we add more detail as to what goes on within those places, because they're so important potentially for this MRSA transmission. So we have special models for those. Anyway, colonized or infectious agents either are present in a place, in which case then the agents just transition themselves through their disease states, probabilistically. Or if there are colonized or infectious agents present in a place, then we have a contact transmission model which represents the probabilities of that infection being transmitted to other people at that place and time. And then we simply, then after this loop occurs, we log all the disease state changes for every single person or agent in the model at that hour. We advance the hour, and then we keep going through that loop until we reach 10 years. So one of the things that should jump out at you is that the logging of the disease states creates a very large amount of data. So we could reconstruct where every single agent was and with who else every other agent was at every hour in the specific place for a period of 10 years. So this is a lot of data. And the reason that I'm mentioning that is I'll kind of talk about, this idea of simulation analytics on model output later. Anyway, I talked about the jail, and as I said, we have a more detailed jail model in which, if we look at the community, which is the city, so to speak, we represent people who are going to jail, probabilistically, based on real data, by zip code and socio-demographic characteristics, who, for the most part, go to Cook County Jail. It's the largest jail in the U.S., actually. So we are ignoring the other jails right now. But in any case, and then within the jail, when an agent gets to jail, they have various contacts with other agents who may or may not be infected with MRSA. And as I said, the reason for doing that is because it is thought that there would be a higher concentration of people in the jail who have MRSA, just because of the communities that they're coming from. So that, by going through that process on an hour-by-hour basis, then it allows us to simulate these timelines for each person in the jail, and to represent what their disease states are during this timeline for the disease states of interest. So that process allowed us to record how many people were actually getting infected in the jail based on all the best available information. Considering how many went into the jail. In addition, we modeled the hospital, what I would call the hospital workflow, relative to people who are entering the hospital from the community, which is this little node, and being assessed. These are the various steps through, the hospital process for either being admitted to the hospital, assessed for whether they're at high risk or low risk, which would introduce extra screening and testing, etc. And all the ways that an agent who enters at one of these places, either entering the hospital or this little bubble represents an agent who becomes infected in the community, doesn't go to the hospital. And all the steps in the process and the interactions of people who are being infected with the community, with people who are actually working their way through the hospital system. And I'm not going to go through all the detail, but the one thing to look at that this diagram allows us to do is to model a decolonization program in which people who have presented themselves at the hospital and who are tested and found to be positive for C and MRSA have an option of having their entire household decolonized, which consists of, kind of advanced scrub downs and things over a period of 4 weeks, to reduce the number of people that are actually colonized in society. And this is a program that was actually instituted in the Netherlands on an experimental basis. It's probably not possible to implement this in the US, but the model allowed, because of, the way that, people like their privacy and all that, but, and those issues. But I think that, the question is, what if we did? What would be possible? Would it be possible, how big a dent would it make in the population of people who are colonized with MRSA? Those kind of issues that can only be addressed by using a model of the process. And this whole decolonization strategy is what I would call a grand intervention. But anyway, this is getting to results from our MRSA modeling for, oh yeah, I forgot to put the grand number publication in, sorry. Yeah, I was rushed this morning. But anyway, what we have, here's a graph that shows several things. In red, if this is red, this line here represents the actual estimated infection incidence of CA-MRSA in Chicago, between 2001 and 2010. So this is the historic buildup of the epidemic, as the data suggests. There's a lot of error band on that estimate. The yellow band is around the red, based on the estimation. So we had separate studies going on to establish these curves for Chicago and other major cities through a meta-analysis. And then the bluish line here, the bluish band, represents the model output over the range of the variability and uncertainties that we included in the simulation itself. So what this process, so anyway, the fit is pretty good. The fit is pretty good. And what we had to do to make this fit is a process I would call extensive validation, not validation, it's calibration. So we're calibrating this giant model with these millions and millions of variables to one data trajectory, and trying to find the transmission parameter values within the large ranges of uncertainty for those parameter values, such that we get a good fit. Okay? So this was quite an extensive process. And the reason I'm mentioning that is because I might get to that in the next 5 minutes to talk about challenges for this kind of agent-based model calibration, as I would call it. Not validation. Much different thing. Anyway, the model here, the model also allowed us to reproduce the patterns within zip code areas of the buildup of the number of people that were presenting themselves with CA-MRSA infections as well. And that's what those little red circles, I don't know, there's an animation, let's see if it works today. Okay, it doesn't work today. That's because of the projector. They kind of. So anyway, so looking at the model results that came out of the full simulation and plotting it against the actual observed data, the fit was very good, without any extra calibration or anything. So this at least is suggestive that the model is. And based on the paucity of assumptions within the model, that the model is maybe a reasonable tool, at least to compare against itself for what the effects of interventions are, and possibly represent the real world. But we do have to be careful when we're talking about things like validation and things like that, because we're not like uncovering the basic laws of the universe or anything with these models. We're more like fitting them to what makes sense and what seems empirically comparable based on some reasonable theory. That's out there. Anyway, the model is also good, as you can imagine, for producing all kinds of other data that's very useful. So here we have all the places that we were looking at, and the model is producing the contact patterns for people by age group for each place. So it reveals a lot of interesting patterns. There's a lot of people, for example, in jail between 19 years and 44 years of age that are actually having contact with each other in that same age group, things like that. And in part, this helped us figure out what the ranking is really of where CA MRSA is most likely to be transmitted by place type, as well as age group and things like that. Some future work, which really would stem from looking at doing a lot more work with the log files, has to do with contact networks. And if you followed Ebola or other outbreaks of big diseases, people are always, the first thing they do is they deploy a set of people to recreate a contact network. And they're trying to find out patient zero, who had contact with who when. And they work that backwards, and they find, the little boy in Guinea who, ate the dropped fruit from a fruit bat, which transmitted the Ebola virus to a human, and as the first case, and started everything, that kind of thing. So anyway, the model eventually will allow us to figure out, in a simulated world at least, what these potential, agent or contact patient zeros are as well. Okay, so this is actually the animation for Chicago, but that doesn't work either, I guess. Okay, so just imagine how this animation would be working. It works on my computer, believe me. So here we have activities. Here we have Chicago. These are the columns represent various places, residents, businesses, or schools in Chicago. So for a period of 1 week, we will be animating the number of people, which is designated by the height of the column, at each of those places. So it's kind of a cool animation. I wish you could see.
So
And
we have just under 10 minutes left.
Yeah.
Do you want using time for questions? Do you want to talk to the end?
Well, that's a good question. I'll think about that. And then over here, we can plot as well in the animation each agent and who if they were infected or colonized, who they were colonized by in the last period. And so people move around, so there's a little line between them, things like that. It's a great animation. You'd love it. Okay, so I think that I do want to have some questions. I won't talk about Ebola or other projects, other than to say we are, in some of our other projects, we are combining networks, estimated networks, egocentric networks, things like that, combining into local networks, and working those networks into the relationships that we're modeling within an agent-based modeling framework. We have a case of a project in HIV modeling, as I mentioned, in which those networks then would indicate who partners are and kind of allow us to propagate disease through those networks potentially. Then we have another kind of project where we're looking at community health information that's being passed to the community, tracking that, surveys as to see who tells who what, and kind of create information networks that way, and then modeling that whole process within an agent-based framework, time stepping through the whole process. The networks are changing potentially, adding or not. So the challenges, there's many challenges. We need better synthetic populations that model activities, and a real challenge is to do better behavioral models of how people actually behave. There's a lot of interesting, behavioral literature, and we all know about behavioral economics and stuff like that. But that information, in particular from behavioral economics, is usually not in a form to make a behavioral model. And so that's a great research area. We'd like to incorporate real-time data feeds to update model state. And whether that comes from sensor data that's coming from sensors being deployed like in Chicago and other places, or from social media data that people are doing a lot of work in. That would be really interesting to kind of calibrate a real-time simulation that's continually updated according to what's going on in the real world. And then finally, an area that I really didn't talk too much about, which is a primary interest of ours, is large-scale computation, which can really benefit from ensembles or really large numbers of distributed simulations that are going on. And what we'd like to do in the future, of course, is to do a lot of uncertainty analysis. Do social forecasting, similar to like weather forecasting, where there's not just one outcome, but it's a range of possibilities. And the range itself is scientifically plausible based on all the inputs and assumptions we've made. We'd really like to get to intervention analysis. What would be the effect of an intervention, as simulated in our model? And finally, how can we make the social systems better perform according to, being more effective and efficient? And then there's a big area of simulation analytics that we like to address, in which, these huge data sets that come out of agent-based models, which contain all the information you could possibly ask for, need to really somehow be, both logged and then systematically mined, like in data mining almost, for the hidden relationships and other information that's, just couldn't be identified in a manual process. So with that, I'm done. Is there time for
a couple quick questions? So, go at it, guys.
Okay, who's first?
Well, since I only see two, you go first, and then you go.
Fair enough. So I had a question with regards to the data you're tracking about the interactions.
Yes.
Do you only track where agents are and thus who they're co-located with, or are you actually tracking their behavior at that point? So are they in contact with someone who's actually infectious, or can you track that process as well?
Well, we can track if they're in contact with someone who's infectious.
Yeah.
Based on their co-location. Yeah.
You could track whether there could be an infection. Do you actually track whether there is an infection?
Yes.
Okay.
Yeah, we track that. It's just a concept that I refer to agent state. Whatever agent state that's in the model, we can track what all the agent states are.
Yeah, but for example, if I'm co-located with 3 people that are infectious,
Yeah.
and I get infected, then what does this tell me? Who has infected me?
Well, yeah, that's a good question. And that's particularly difficult for, doing this patient tracing thing. So we're looking at what the possibilities are, keeping all those 3 people on the list of possibilities.
Okay.
And seeing how that, works out as well. But contact tracing people in the real field have that problem anyway. So, yeah.
Okay, so.
I think Ray had a question, so I'll yield to him. Oh, he didn't? Oh, okay.
You didn't have a question?
No questions. So I'm a little bit unclear of the specifics of how you modeled all the interactions between people. So basically, when they move between a location, except for the jail and the hospital, is that sort of, I don't want to say just a proxy, but is it a proxy for basically moving them into their own little container and then only running the probabilities of infection on them?
Yeah.
And then, so basically moving people between containers.
Yeah, that's one way to look at it.
Okay. No, I was just curious because.
Yeah, it's so we could start out with some model that has like the usual compartmental models for a disease. We could just have 3 boxes, and those are the compartments. So another way to look at the agent-based model is we're just bringing all those compartments into compartments that are defined by place, in addition to the activity they're engaged in at that place. But then all the agents that are there have a state,
And so the people that are in these different places, do they have particular behaviors that are tied into that place?
Oh, yes.
Okay.
Oh, yes, of course.
Okay.
They know where they are. I'm in the hospital, so I want to behave this way.
Okay.
Or I just heard about, there's an Ebola, outbreak or something, so I'm going to evacuate Chicago.
Okay.
That kind of thing. Yeah,
Cool.
potentially. Okay, I guess they're
No,
looking
one
to
more
get
question.
one more.
Are you meeting with him later?
I don't have a meeting with
Okay, well, maybe you can just slip into mine and ask
Okay.
them. So we're gonna go
Sure.
this one.
One more.
So obviously, like the parameter space is really huge. You're making
Yes.
lots of assumptions and lots of things like that. Like it seems like when you're talking about model fitting, it's a matter of finding one set of parameters, one vector of parameters that matches well enough to the curve. But
Yes.
it's fair to say that there's lots of different sets of parameters that could.
We haven't
fit.
found it that there's, you would think so. You would think, there's an infinite number, large finite number of degrees of freedom, so, it's just arbitrary. That's not what we found, at least with this model. Because the model itself is so tight together internally, there aren't that many degrees of freedom. I could have showed you another graph where we looked at the combinations of parameters in which the epidemic never occurred. The transmission was too low, it just died out. Or how it became a super epidemic. And, my interpretation is, maybe those were possibilities that didn't happen, as opposed to parameter values that were wrong. So, it's a great question, and it's a great to think about what the meaning of the model underneath are. Yeah, thank you for that.
Thank you.
All right. Thank you everyone for coming. We have to-